transcription and translation. Kita berkenalan dulu dengan the main ingredients for this transcription process. Okay, we have RNA. Okay, it is a single strand. We already know RNA is a single strand of polynucleotide, and very important because they involved in protein production. Okay, to produce protein. There are three types: mRNA, rRNA, and tRNA. mRNA is messenger, pengutus. Okay, sebelum ni saya ada mention messenger, pengutus. Dia carry information. Dia carry information yang dia information nanti akan ditranscribe and translate into protein. So mRNA is a linear sequence. Yes, it's a single strand. So it is a linear sequence that copy information, whatever information contained in DNA or genes. Okay, remember kita ada banyak, kita punya DNA dia punya million tau sebenarnya, million. Okay, cuma dekat situ adalah gene 1, gene 2 that control certain character kan. Okay, so mRNA is a single sequence. Okay, and when you translate you get protein. And rRNA, R stand for ribosomal RNA. Okay, R stand for ribosomal RNA. So, nanti you akan belajar tak ribosome ni, kalau you perasan lah, dia bentuk dia macam ni, ribosome. Consists of small and large subunit. So, ribosome ni, the main ingredients to make this ribosome is rRNA and also proteins, a lot of proteins to make this subunit. Okay, to make ribosome, you need rRNA and also proteins. Okay, so rRNA is component of the ribosomes, also involved in protein synthesis. Next, we have tRNA ataupun nama dia transfer. Okay, transfer RNA. Maksudnya dia transfer kan kelas. Dia transfer apa? Dia transfer amino acid. Okay, transfer amino acid to ribosomes. Nanti you akan nampak. Okay, kejap function dia. Sehari tu saya dah explain tentang codon. Betul kan kelas? Saya dah explain tentang codon kan?
Ada.
Okay, so kelas you dah okay sikit. Okay, so 5', 3', imagine this is mRNA. And you have this codon, for example, you have A, U, C, G, A, U, C, A kan? U, A, whatever lah. So yang ni dah ada mRNA, so you take note sebab tu saya ada U dekat situ. No thymine because this is RNA. And this is codon. Saya dah mention itu kan. Codon. Every set here which consists of 3 bases dekat sini, kita panggil dia codon. Okay, now tRNA saya nak lukis macam ni, tRNA. Macam penyapu sikit. tRNA ni dah ada satu site tau. Site ni untuk amino acid attachment. So amino acid akan attach dekat sini. Dekat sini pula dia ada base tau. Kita ada base yang akan bind kat sini. Kita panggil dia anticodon or anti-codon. Okay, Mak Saleh dia anti, dia slangkan dia lah. Anticodon. Okay, so AUC for example. So codon will complement with anticodon. So kalau codon ni A, sini mesti apa? U. Kalau U mesti A, C mesti G. So dia akan bawa kat sini. You boleh imagine tak tRNA kat sini. U, A, G. Ni amino acid. Dia carry amino acid. Sebab tu tRNA function dia tulis apa kat tadi? Dia kata amino acid function dia carry specific. Okay, specific amino acid. Okay, to ribosome. Okay, dia carry sebelum kepada ribosome tu dia bawa kepada mana? Sebab nanti dia macam ni tau kelas. mRNA tadi dia akan ditranslatekan. It will be translated in ribosome. Ni mRNA this is mRNA. Dia akan ditranslatekan oleh ribosome. Okay, so nanti tRNA nanti dia akan datang dekat ribosome ni, dekat area ni. Nanti kita akan belajar proses tu. So tRNA datang, gedik datang kat sini kan, and dia akan carry amino acid. Okay, sebab dia tidak kata tRNA carry specific amino acid to ribosome. Actually dia carry, dia akan complement with anticodon. So dia akan complement with codon in mRNA yang dia dah bind dengan ribosome. Okay, sebab previously you belajar kan fungsi ribosome. Okay, you dah belajar fungsi ribosome masa belajar cells. Ribosome synthesis protein, you kata. Synthesis protein kan. And you dah boleh imagine ribosome, lokasi dia dekat mana? It is located in RAF, ER, kan? And then dia ada juga free ribosomes. And ribosome is located in cytoplasm. We can conclude like that. Okay, in ribosome, in cytoplasm. Macam tu lah. Panjang pula penerangan dia. Cuma saya nak cakap rRNA, RNA ada 3 komponen. mRNA, rRNA and tRNA. So semua RNA ni involved in protein synthesis, in production of proteins. Messenger, copy, information. rRNA is a component of ribosomes and tRNA transfer specific amino, betul kan? Transfer specific amino acid to ribosomes. Clear? Function sahaja tu. Okay?
Yes.
Next. Okay, thank you for the feedback. Okay, now kita nak tengok, okay, basic principle of transcription and translation. I'm sure you dah tahu dah benda ni. Okay, transcription, the synthesis of mRNA, ya lagi specific. The synthesis of mRNA using DNA template. Okay, the information is transcribed, is copied into mRNA. Okay, so it will produce mRNA. After transcription, dia akan proceed with translation. Okay, tak perlu hafal macam transcription lead to the translation. No need to memorize. You dah tahu dah. After transcription, we go to translation. Translation where you want to translate the information from mRNA into protein, into polypeptide. Okay, and kat mana dia berlaku? Dekat ribosome. Ribosome are the site of translation, tempat berlakunya. Kalau nak jawapan lagi tepat, dia berlaku di ribosome, dekat cytoplasm. Tu lagi complete lah jawapan dia. Okay. Now, kita tengok, okay, transcription in prokaryotic cells. Okay, for your information class, okay, selalunya if we have any study about DNA, we will start with bacteria. Why? Because bacteria easy to duplicate. I mean, dia senang kita nak growkan dia dalam medium and dia akan membiak dengan cepat. Compare kita nak buat kajian dekat manusia, it takes time untuk produce offspring kan, lambat proses tu. Okay, so normally we use bacteria, okay, prokaryotic cell to see the concept of DNA, genetic, semua kan. Okay, now in prokaryotic cell, transcription. Okay, dia cakap transcription occurs in cytoplasm. Okay, why in cytoplasm? Because prokaryotic cell, example bacteria, dia tak ada nucleus, no nucleus. Remember? No membrane bound organelle. So transcription berlaku dalam cytoplasm. Okay, so what happened here? You can see here, DNA will act as a template. Okay, they provide instruction to synthesize mRNA and after that translate into polypeptide with the help of ribosomes here. And here yang panjang ni, this is mRNA. Ribosome akan translate semua informasi, all the information in this mRNA into polypeptide. Okay, how about translation? Okay, the information encoded in mRNA determines the sequence of amino acid to form a specific polypeptide. Ayat dia memang panjang dan susah. Again, you ingat lagi saya cakap tadi, setiap codon ni dia akan specify for one amino acid, another amino acid. When you join them together through peptide bond, okay, peptide bond, you get polypeptide or you get protein. Okay, ayat je belit-belit. Okay, so this specific. So you get the sequence of amino acids. So this is a sequence of amino acids. Okay, dia akan form specific polypeptide. And it occurs at the ribosomes in cytoplasm. Nampak? Ni jawapan yang lebih complete. Occurs at the ribosome in cytoplasm. Faham semua? Ada masalah?
Faham. Faham.
Okay, good. Okay, kalau faham saya proceed. Now, how about transcription in eukaryotic cells? Okay, ini tadi flow dia tadi, you dah familiar dah from DNA to mRNA to protein. Okay, transcription occurs in the nucleus for eukaryotic cell because eukaryotic cell they have nucleus. Okay, they have nucleus. So transcription occurs in the nucleus because DNA inside nucleus. So when you transcribe DNA, you get pre-mRNA. Dia lain, dia tak dapat mRNA straight away tau. Dia akan dapat pre-mRNA first. Okay, this is macam dia dapat pre-mRNA and then dia akan undergo RNA processing and produce mature mRNA. So kita nak mRNA, we don't want pre-mRNA. But tapi dalam eukaryotic cell, it is slightly complex lah. Okay, and then gene dia punya size dia lebih besar daripada bakteria. Okay, so you get pre-mRNA, okay, and a gene provide instruction for synthesizing mRNA. Of course lah, ada bahagian gene. Yang gene DNA ni lah yang akan provide instruction lah untuk penghasilan protein actually. Okay, ini general, ayat general sahaja. So what is RNA processing? Dekat sini kata, the original RNA transcript or pre-mRNA is processed, is modified. Dia akan proses, modify sebagai mRNA lah dulu sebelum dia jadi mRNA, functional mRNA. Nanti kita akan baca macam mana dia modify. Okay, now next, translation. Okay, for eukaryotic cell. Information encoded in mRNA determine the sequence of amino acid to form a specific polypeptide. Again, maksud dia sama je. Setiap codon tadi akan specify for one amino acid and dia akan form specific polypeptide. Okay, and it occurs at ribosome in cytoplasm. Nampak tak perbezaan dia dekat sini? Okay, in eukaryotic cell, transcription occurs inside nucleus, but translation sama macam prokaryotik, berlaku dalam cytoplasm. Okay, apa lagi perbezaan dia? Perbezaan dia kalau you tengok dekat sini, dia akan straight away produce mRNA, but for eukaryotic cell dia akan produce pre-mRNA and dia akan undergo RNA processing to produce mRNA. Tu perbezaan jelas, okay, clear differences between eukaryotic and prokaryotic cells. Okay, now next. Tarik nafas semua, saya agak laju juga ni, okay, sebab takut tak sempat nak habis. Semua okay lah? Okay.
Okay, madam.
Now, genetic code. Okay, for each gene, okay, okay, ni banyak gene dekat sini. Ya, gene dekat sini kan. Because we know that chromosome kan, ni strand dia ni million tau. Kalau you buka dia punya strand tu, million dia punya panjang dia tu, size dia tu. Okay, and ada banyak gene dekat situ kan. I don't know what kind of gene yang control apa kan. Banyak trait yang gene control. Okay, for each gene, one DNA strand function as a template for transcription. Okay, we know that DNA template, DNA is double strand. Okay, we have 5, 3 and 3, 5. But only one serve as a template. Hanya satu je yang bertindak sebagai template. One only serve as a template. And for your information, kan kita tahu mRNA yang saya tulis tadi kebanyakan saya buat 5, 3 kan. 5 prime, 3 prime kan. Maksudnya kelas, apa dia? Maksudnya satu je jadi template. Template dia apa? This is mRNA. This is single strand. Kalau single strand mesti single template. So kalau ni 5, 3, template dia mesti 3, 5. Okay. So 3, 5 will act as a template strand. Okay, 3, 5 will act as a template strand. So take note ni. Nanti masa lab nanti Dr. Mazhar ni tanya, yang mana satu nak jadi template strand. So you tengok 3 prime, 5 prime will act as a template strand. Puan Mazhar ni dia suka tanya tau. Kalau dia tanya, you please respond to her. Bagi feedback. Kalau you bagi feedback, dia seronok sangat. Kalau you tak bagi feedback, dia merajuk. Dia memang dia akan merajuk. Dia akan bagi tahu kat saya nanti. Sebab saya tahu dah saya rapat dengan dia. Okay. So 3, 5 tu is a template strand. Okay, so dia akan guna 3, 5 ni as a template strand and dia akan produce mRNA. And you see here, this is 5, 3. Dia punya partner strand dia tu, 5, 3. Okay, so kita panggil dia non-template strand. Okay, template strand versus non-template strand. And class, nama lain bagi non-template strand is coding strand. And siapa ada idea kenapa kita panggil dia coding strand? Why we call this strand as coding strand? Ada tak idea? Siapa nak sumbang idea? Okay, bukan sumbangkan fakta sumbangkan fakta pun acceptable lagi lah. Okay, agak-agak kenapa nama dia coding strand?
Kodon.
Codon. Menarik juga. Codon. Coding strand. Okay, for your information class, good try. Coding strand ni, kalau you perasan dia punya strand dia sama macam mRNA strand. Nampak tak? Coding strand, macam it give you information juga dah dekat situ. So that's why kita panggil coding strand. And sometimes kalau jumpa sense strand. Sense strand, coding strand refers to non-template strand. Okay, kenapa? Sense, make sense, coding strand, codon. Sebab dia punya strand ni sama dengan mRNA. Except base timing is replaced with U. See? Nampak tak? Ni T replace dengan U. Okay. Nampak tak? Tu je perbezaan dia. So that's why you guna template 3, 5. So dekat sini you ada A, right? So this will be U. This is C, G. C, G, A, U, A, U, A, U. Nampak tak? C, G. So 3, 5 ni dia akan produce mRNA which is sama juga dengan coding strand tu juga sendiri sebenarnya. Okay, sama dengan coding strand except base timing replaced by uracil. Okay, tu je perbezaan dia. So that's why kita panggil dia juga coding strand or sense strand because they have the same strand like mRNA yang they produce too. Okay, so again, please use 3, 5 as the template strand. Okay, so maksudnya your mRNA is produced in a 5-3 direction. Sama macam DNA replication. Remember DNA replication? Direction dia ingat tak? The newly synthesized strand must be 5-3. Okay, sama juga dalam kes ni transcription, the mRNA tu dihasilkan in a 5-3 direction. Okay, yang ni saya rasa you faham dah kan yang kodon-kodon ni? Remember yang the table tu? Okay, mRNA, UGG stand for tryptophan. Kalau nak tahu betul ke tak, you tengok UGG. Okay, dengan U, senang nak tengok. Okay, kita tengok dulu contoh. UGG dengan U. UGG, yes, tryptophan. U sini. Okay, U. PHE, phenylalanine. You jangan hafal gambar ni, jangan hafal table ni. If dia keluar dalam exam, dia akan bagi you table ni. You jangan hafal. Okay? Contoh apa lagi you boleh tengok dekat sini lah. You can try on your own lah. GGC, right? So you pun buat G. Sekejap, dah tak. Sekejap, sorry, pen saya. Sekejap ya. So C, right? C here, sini, so GGC. So you get this glycine, GLY, glycine. Okay, so you ada dekat sini. Okay, so nampak kan? So when you combine this amino acid through peptide bond here, okay, peptide bond, you get polypeptide or protein during translation process. Okay, so I'm sure yang ni you dah boleh buat lah, you boleh pairkan dia kan? Okay, if you have this DNA template strand, this is the RNA codon. Make sure you tahu. Kalau you buat kerja ke, mesti ada direction. Okay, you have to show the direction. So dekat sini, okay, class, saya nak highlightkan perkataan. The term codon it is also be used for the complementary DNA based triplet on non-template strand known as coding strand. Ayat ni akan mengconfusekan you, so no need, you tengok kat sini je. Non-template strand also known as coding strand. Itu je. Sebab dia coding strand, tadi saya dah kata dia sama strand dengan mRNA. Nama lain bagi dia sense strand. Okay, sense strand. So kalau you jumpa dekat sini, nama lain bagi dia apa dia? Non-coding strand or anti-sense strand, anti-sense strand. Faham?
Faham, madam.
Faham semua ya? Diam je, tak ada setuju lah tu ya.
Faham.
Faham, madam.
Faham.
Okay, good. So you dah buat lab dah tau sebenarnya. Okay. Dah ready dah. Okay, now I want to explain about feature of the genetic code. Okay, yang ni this is during translation nanti. Untuk begin the process of translation, nanti you akan ingat ni ya. Dia akan start dengan dekat mRNA tu, dia akan start dengan codon AUG. AUG kita panggil dia start codon. So translation akan bermula di sini. Dia akan cari codon ni, so dia akan mula dekat sini to start the process of translation. Okay, initiation codon or start codon, AUG. To start the process of translation, terminate, dia akan tengok kalau dia jumpa this codon, dia akan stop the process of translation. Yang ni jangan risau, kita akan belajar sekejap lagi. Okay, and then codon of mRNA is read in a 5-3 direction. Saya dah mention dah tadi sebab kita guna template 3-5. So you will get this 5-3 direction. Okay, contoh macam ni lah. Non-overlapping. Each base in the sequence is read only once. So setiap codon tu dia baca hanya sekali. Dia takkan baca banyak kali. Unambiguous. Each code has only one meaning. AUG maksud dia match je. Dia tak ada maksud lain. Tak ada AUG jadi serine ke, jadi tyronin ke, dia tak ada. Dia ada one meaning only. Okay, and then redundancy. Some amino acid are specified by more than one codon. Ada satu amino acid tapi ada banyak codon. Example here, you can see the table. Okay, for example here, ada banyak codon, but the codons specify for one amino acid only. Tu maksud dia. Many codon but specify for the same amino acid. Okay, universality. Okay, the same genetic code applies to all organism. Maksudnya kelas apa? You can use this table, okay? You can use this table for bacteria, for human, for plants. Maksudnya kalau dalam bakteria CAU is his apa ni? Sorry, kita yang mudah ni. Okay. AGU SER, serine. Okay, dalam bakteria serine. Dalam manusia pun AGU serine juga. Okay, dalam plant AGU pun serine juga. Maksudnya the same codon code for the same amino acid in all organisms. Okay, and you can see here kita ada 4 bases kan dekat sini, UCGA RNA, right? So kalau you buat combination dia ada 64 different codons dekat sini. So you boleh kira lah kalau you nak tahu. Okay, we have 64 different codons. Okay. You boleh tarik nafas, geraklah kaki kiri ke kanan ke kiri ke, nak pusing badan. We are about to learn about transcription process. Okay, semua ready ya? Okay, transcription. Okay, you can see here we have 5-3-3-5 dekat sini kan. So it's a double strand. But remember only one strand will serve as a template which is 3-5. And you can see here 3-5 dekat sini. Nampak tak 3-5 dekat sini? 3-5 is used as a template. This is RNA transcript. Okay, now transcription ada 3 stages. Initiation, elongation and termination. For initiation, permulaan dia, initiation, first, okay, you can see here, this is RNA polymerase. Okay, RNA polymerase sama function macam DNA polymerase. Ada beza dekat situ. DNA polymerase add nucleotide. Remember during DNA replication. Sama juga. RNA polymerase akan add nucleotide. But dia ada extra job. Dia boleh unwind the helical structure of double helix. So dia akan unwind dulu the helical structure and then after that dia akan start adding nucleotide. Start the transcription process. So apa yang berlaku dekat sini? Okay, if you notice, this is promoter. Okay, promoter is a region. Promoter adalah satu kawasan where RNA polymerase bind. So RNA polymerase bind akan bind dekat promoter region. Yang ni adalah RNA polymerase. Dia akan bind dekat promoter region. Okay. So setiap gene ada promoter dia sendiri kelas. Okay, for your information, every gene has their own promoter. Dekat promoter tu ada start point. Okay, start point to begin the process of transcription. Kalau you tengok dekat kawasan promoter ni ada start point kan? So dia akan mulakan transcription process daripada start point tu. Okay, pernah tahu kawan saya, dia punya thesis master, okay, master thesis dia punya job, dia tahu apa dia? Cari promoter je pada jin yang dia nak tu. So dia cari jin yang dia nak tu, dia nak promoter region tu, dia nak promoter punya kawasan. Okay, so promoter ni adalah tempat di mana RNA polymerase bind. Okay, so for initiation, RNA polymerase binds to a promoter where the helix unwinds and transcription starts. Okay, RNA polymerase dia akan unwind the helical structure and start the transcription process. Next, elongation, pemanjangan. In Malay kita panggil pemanjangan. Maksudnya apa kelas pemanjangan? Kalau ingat function DNA polymerase 3, dia akan continuously adding nucleotide kan? Add nucleotide. Sama juga. In elongation process, RNA polymerase move downstream. Maksudnya this is upstream, this is downstream. So dia akan move towards this direction. Okay, RNA polymerase move downstream, unwinding the DNA. So you can see here the unwind, the DNA structure unwind. Terbuka dekat sini. And elongating the RNA transcript in a 5-3 direction. If you see this is 3 kan? So RNA transcript akan start dengan 5. Dekat hujung ni adalah, sini hujung ni adalah 3. So dia akan elongate in a 5-3 direction. So lepas dia transcribe this area, dia akan reform a double helix. Maksudnya kau you nampak tak dekat sini? Hanya this area je yang akan unwind. Yang ni dah apa ni? Dia dah transcribe, so dia akan rewound balik. Okay? Dia akan form double helix structure balik. So dia akan elongate. Elongate. Maksudnya RNA polymerase akan continuously adding nucleotide in a 5-3 direction. Okay. Next we have termination process. Termination, apa maksud termination? Stop. Okay, stop the process. Okay, RNA or mRNA transcript is released during termination. And after that, polymerase, ni RNA polymerase akan detach from the DNA template. So the process stop. So that is transcription process. Semua clear ke walaupun saya ajar laju?
Madam,
Ya.
yang RNA polymerase tu dia add nucleotide, RNA nucleotide lah kan?
Dia add RNA nucleotide sebab dia nak buat RNA.
Okay.
Okay, kalau you nak be more specific, you boleh cakap RNA polymerase add RNA nucleotide. Okay, RNA nucleotide, which is good kalau you jawab dalam exam spesifik RNA nucleotide. And then RNA polymerase ni sebenarnya dia ada spesifik lagi, RNA polymerase 2. RNA polymerase 2. Okay. Ada soalan tak dekat sini? At least nampak tak summary dia?
Madam,
Ha.
jadi lepas dah complete RNA tu, RNA
Sekejap.
polymerase akan keluar lah. Keluar daripada,
Yes, dia akan detach. Sorry, tak mana tadi. Dia akan detach. Ada soalan lagi tak? Dia akan detach lah daripada tu. Kat sini tadi kan? So dia akan detach kan? Detach means dia keluar lah, exit. Nampak tak? Idea sikit pun nampak. Semua okey? Saya tahu tengah hari.
Okay, madam.
Okay. Lagipun you mengadap laptop ni penat kan? Dia punya inilah. Okay, tapi tak apa. Saya pun I have 2 hours class tadi, so penat juga. Okay. Now, tadi summary class ni baru kita nak pergi detail sikit je. Okay, tapi jangan takut, nampak menakutkan. Tapi tak susah pun, senang sangat. Okay kelas, tadi saya ada mention promoter region. Okay, remember promoter apa dia? It's a place where RNA polymerase bind. Jangan baca lagi tengok dulu. Promoter is a place where RNA polymerase bind. Macam promoter dekat yang jual produk. Promoter buat apa? Dia promote kan, datang beli produk saya kan. Promoter, sama juga. This promoter, okay, you tengok gambar ni je sekarang ni. This promoter akan promote, dia akan attract RNA polymerase, nampak? Yang ni dia more specific, dia bagi RNA polymerase 2. Okay, akan bind at promoter region. Now, dekat promoter region tadi saya ada mention dia ada start point to begin the process of transcription. And nampak tak template strand here, which is 3, 5, the template strand. And dalam promoter region ni dia ada tata box tau. Nama dia tata box. So those yang buat research on promoter, dia cari promoter region, promoter gene. So mungkin dia akan tengok ni, ada tak tata box ni? What is tata box? It's a short sequence about 25 nucleotide that consists of thymine, adenine, thymine, adenine, thymine, adenine, adenine, thymine. Okay. So bila dia ada banyak TA, so kita panggil dia tata box. Okay, so tata box lokasi dia dalam promoter. So those yang cari promoter, yang buat kajian, nak cari promoter, clue yang pertama, carilah tata box. Okay. Sebab dekat situlah bermulanya transcription. Okay, dekat sini start point dia. Okay. Okay, next kalau you tengok, tadi saya ada mention RNA polymerase bind at promoter region. But before RNA polymerase 2 tadi bind at promoter region, ada benda lain dulu yang akan bind. Siapa dia? Transcription factors. So transcription factors ni is a protein actually. It's like macam tourist guide tau. Dia yang akan bind dulu dekat promoter region, barulah RNA polymerase can bind to this promoter region, forming transcription initiation complex. Sebab kompleks dah. Sebab you ada transcription factors, you ada RNA polymerase 2 kan. So kita panggil dia transcription initiation complex forms. Okay. So apa RNA polymerase buat? Tadi saya dah mention RNA polymerase 2 akan unwind the DNA double helix. And then dia akan begin the process at start point on the template strand. Dia akan add RNA nucleotide in a 5-3 direction. Sebab template yang kita guna 3, 5. So if this is C, this is 5, this is 3. Maksudnya RNA polymerase akan continuously add nucleotide to the 3 end here. Faham tak? And dia akan elongation process. Faham semua?
Faham.
Okay. Thank you. Okay, sebab respon. Next, kita tengok elongation. Kalau you dah faham tadi, dah tak ada masalah dah. And you can see here, this is a template, 3. Okay, here. So this is a template. You can see here, dia start dengan 5 lah kan. 5 and 3 ni, ada 3 end dekat sini. Dia akan, RNA polymerase will continuously adding RNA nucleotide one by one here. Nampak tak? One by one. Okay, dia akan elongate. Continue to separate the DNA strand. Tugas dia, RNA polymerase. Okay, and dia akan add RNA nucleotide to the 3 end here. You can see here the 3 end here of the growing RNA polymer according to the base pairing rules along DNA template in a 5-3 direction. Okay, bahagian belakang ni pula kelas, lepas dia transcribe, dia akan form double helix balik The double helix reforms and new mRNA tadi, RNA molecule pulls away ataupun RNA transcript tadi, dia akan detach from its DNA templates. Okay, that is elongation. Next process, last sekali, transcription. Okay, transcription, what happened during transcription in bacteria, okay, dan juga in eukaryote. So bacteria ni prokaryote lah kelas. Okay, prokaryotes. Termination ni proses dia nak stop. Okay, stop the process of transcription. Okay, apa yang berlaku if you have this mRNA sequence, okay, dalam prokaryote, RNA polymerase transcribe a terminator sequence in DNA. Cuma saya tak nyatakan what is the terminator sequence. Kita tak belajar detail Mungkin dia jumpa something terminator sequence dekat sini, satu sequence. We don't know apa dia punya sequence, dia tak diterangkan kat sini. So bila dia RNA polymerase, dia gerak ke depan, dia jumpa this terminator sequence dekat sini, dia akan stop the process. So dia akan stop transcribe. Okay. So the transcribe terminator sequence on mRNA tadi signal the end of transcription. So what happened? Polymerase detach from DNA, okay, and release mRNA. And then after that barulah dia translate without further modification. Okay, tadi saya dah mention perbezaan pertama tadi, in prokaryote, you don't modify mRNA. You memang dapat mRNA. Straight away you dapat. Mana tadi? Kan? Daripada ni tadi. You straight away you get the, apa ni? Dekat sini. You get mRNA here. Okay? You can see you get mRNA dekat sini. No need to translate. But for prokaryotic cell, you need to modify the pre-mRNA into mRNA. Tu perbezaan dia. Okay? Okay, in eukaryotes, okay, RNA polymerase to transcribe polyadenylation signal sequence on the DNA. So, apa jadi dekat sini? Okay, masa dia transcribe area ni, DNA kepada mRNA, dia jumpa polyadenylation signal sequence on the DNA. So, dia jumpa apa dekat DNA tu? Dia jumpa, mungkin polyadenylation ada banyak A. Sebab tu nama dia polyadenylation signal. Meaning that, banyak adenine. Okay, so bila dia jumpa A dekat DNA ni, what happen? Dia akan stop transcribe. Okay, dia akan stop transcribe. So this produce polyadenylation signal AAU in the pre-mRNA. Sebab nampak dia jumpa dekat sini kan? Okay, so dia akan produce polyadenylation signal in the pre-mRNA. And lepas dia jumpa this polyadenylation signal, dia ada signal untuk dia stop the process. So dia akan release 10 to 35 nucleotide past this polyadenylation sequence. Saya ada gambar dia. See here? Maybe this is the start codon. Proses berlaku tadi. This is a stop codon. Start, stop. Yang ni polyadenylation signal. Ada banyak polyadenylation signal dekat sini. A banyak kan? Banyak adenine. So, UTR stand for untranscribed region. Maksudnya dia tak transcribe region tu. Untranscribed region. Yang ni je transcribed region. Okay, region that include protein coding segment. Maksudnya kalau you transcribe this area, you will get protein. Okay, yang ni tak ada apa maksud. Untranscribed region. You put untranscribed region. Okay. And maybe promoter region is somewhere here. Okay, before start. Okay, start codon. So, dia start dekat sini lah. Okay. Faham Okay. Next tadi. Ya.
Yang terminator sequence tu boleh explain balik tak? Tak faham.
Terminator sequence tu dia general sebenarnya. Dia tak nyatakan apakah terminator sequence. Dia just cakap, saya boleh reka je dekat sini. Mungkin bila dia jumpa AUU dia stop. Ataupun dia jumpa AUG dia stop. Tapi saya tak nyatakan apa terminator sequence tu. Saya tak go detail sebenarnya. It's just that RNA polymerase tu dia transcribe. Maksudnya mungkin dia transcribe a certain sequence yang saya tak explain apa terminator sequence tu, tapi bila dia transcribe that terminator sequence, dia akan stop the process. Tu je. Cuma dalam eukaryote dia detailkan lagi. Dekat eukaryote dia bagi tahu terminator sequence bila dia jumpa A yang banyak ni. Okay, ataupun baca AAU, adalah sikit U dekat sini tak kisah, tapi dia ada banyak adenine. So ni adalah signal untuk stop. Dekat sini dia tak ada AAA sequence, tapi cuma dia tak cakap apa dia sequence dia. Dia just cakap in general, when RNA polymerase transcribe terminator sequence, then dia akan stop the process. So kalau you nak tahu apakah terminator sequence yang menyebabkan proses stop, you boleh Google. Cuma yang ni jadi kompleks lah. Dia akan kompleks lah sebab kita tak belajar, syllabus kita tak cover terminator sequence tu apa dia. Okay, tu je. Okay. Okay, now tadi kan you have pre-mRNA, you kena modify, you have to modify, you have to process this pre-mRNA so that it become mature mRNA. Okay, ni dalam eukaryote. Okay, eukaryote, you have to modify. Okay, apa yang dia modify kelas? Saya tak nak you baca ni lagi. Baca akan menyebabkan you pening, you tengok gambar. Modification. Okay, first modification is by adding this one. 5 cap. First modification. This is 5-3 kan? The mRNA yang you dapat tu. So dekat upstream dia, bahagian upstream dia, atas upstream, dia akan add 5 cap. Okay, 5 cap ni is a modified guanine nucleotide added to the 5N. Okay. Kalau you jawab just add 5 cap, you dah dapat markah dah. Okay, add 5 cap. And then second modification, okay, dia akan tambah poly-A-tail. Ni signal untuk berhenti Najan. Tapi dia akan tambah poly-A-tail juga di akhir transcription. Dia akan tambah A. Okay, dia akan tambah banyak A. That is second modification. Okay, faham Dia modified pre-mRNA. And the third modification, okay, saya dah okay. Ni dia ada 5 cap, dah ada poly-A tail, and you tengok dekat sini kawasan ni, this area. This is pre-mRNA. Dia ada intron, and kawasan ni kita panggil dia axon. Intron, this is axon. Okay, for your information, intron ni kawasan yang tidak memberi maklumat. It don't give you information about protein. Okay, tak ada maklumat. Axon adalah bahagian yang ada maklumat. It will give you information about protein. Okay, so we don't want the intron region because it doesn't give you any information. So we just want axon region. So macam mana nak buat? Kita akan cut the intron region. Intron region will be cut and axon region here will be joined together, will be spliced together. You see here? Introns cut out and axon spliced together and dia akan cantumkan dekat sini. Now you have the 5 cap, you have the poly-A tail, and you have the axon region only. That is the modification that occur in pre-mRNA. Okay, dia ada 5 cap, dia ada poly-A tail, and they remove intron region and join the axon region. Because axon region contain information. Okay, but intron region doesn't have any information. Is that clear? Faham ke tak?
Faham?
Okay. Agaknya intron ni ada maksud, ada tugas tak? Mesti dia ada tugas kan kalau Tuhan ciptakan dia. Okay, saya ceritakan saya punya kajian. Okay, ini kajian saya saya ada buat kajian about genetic diversity, kepelbagaian genetics. Okay, saya nak different. Kalau you tengok pokok pisang, you nampak sama je kan pokok pisang. Pisang hutan kalau tepi jalan sama je. But maybe they are from the different species. Daripada spesies yang berbeza. So macam mana nak bezakan dia? So, you boleh tengok dekat bahagian gene dia, ataupun kita panggil dia coding region dia, which is axon tadi. Okay, axon. Ataupun you boleh tengok dekat intron dia. Intron dia. So saya ada bahagian saya punya kajian, saya ada kawasan intron, saya ada kawasan gene which is axon. Saya ada so yang intron ni walaupun dia tidak berguna sebab dia tak bagi maklumat in the form of protein, tapi dia memberi maklumat tentang perbezaan, the difference between one species and another species. Kan dia mudah macam mana dia bagi maklumat. Contoh kalau spesies A, ATT, spesies B dia ATG. Dia ada perbezaan dalam intron tu sendiri. So dia berguna dalam kajian genetic diversity untuk differentiate untuk between one species and another species. Okay, kalau you kaji axon ni, yang ni berguna untuk kajian apa? Gene expression. Maksudnya protein. Protein apa yang dia hasilkan? Tu perbezaan dia lah. So in this case, because kita nak tengok protein kan, so we remove the intron region, so we only have the axon region. Okay, the region ataupun the coding region. When you translate this region, you will get amino acid sequences of polypeptide. Faham semua?
Faham.
Okay, so class, kalau you tengok slide ni, you akan faham. So RNA processing. First, you add 5 cap. Okay, the 5N, dia dekat bahagian 5 tadi kan, dekat 5 prime tadi, saya kata upstream dekat sini tadi. Dia akan receive 5 cap. Okay, and dia akan ada kat sini, dia dekat 3N tadi, dia akan makes a poly-A tail. Consists about 50 to 250 adenine sahaja. Ditambah lagi. It's like macam you pakai topi dan you pakai kasut. Cap tu is like topi. Poly-A tail is like macam kasut. Okay, dia ada depan dan dia ada belakang, bawah. Okay, Di sini dan di sini modification dia. And then RNA splicing tadi. Okay, kita remove non-coding sequences intron. Tak ada masuk, dia tak akan express jadi protein. And you join the coding sequence which is the exon. Apa function dia kelas? Kenapa kita buat modification tu? Sebab kelas, it's like macam bila you pakai topi, you lindung diri you daripada matahari. Okay, you avoid yourself from sun exposure yang panas sangat tu kan. Bila you ada tail dekat sini, it's like macam you pakai kasut tau. You protect you punya kaki. Macam tu juga. Sama juga mRNA. When you modify, okay, you facilitate the export of mRNA to the cytoplasm. Okay, dia membantu. Mungkin bila dia ada poly A tail, dia susah nak degrade tau. Dia lebih stable, more stable. Okay, dia more stable, so dia facilitate the export of mRNA to the cytoplasm for translation process. And then apa lagi? Dia akan protect mRNA tu. mRNA tu daripada nukleus kan, dia nak keluar pergi cytoplasm. So dia akan protect those mRNA from hydrolytic enzymes. Ada banyak hydrolytic enzyme in your cytoplasm. So bila dia pakai cap, dia pakai poly A tail, dia akan tadi more stable and dia akan protect daripada hydrolytic enzyme. And again, help ribosome attach to the 5' end of mRNA. For your information class, 5-3. Dia tak ada dalam nota ni Cuma kalau you baca buku dia akan dekat bahagian sini untranscribed region tadi UTR tadi, dia ada satu tau site. Ni adalah site untuk ribosome bind. Okay, ribosome. Ribosome bind dekat sini. Dia ada satu bahagian dekat mRNA ni site for ribosomes to bind. Dia berada pada kawasan untranscribed region. Okay, ribosome akan bind dekat sini. So help ribosome attach to the 5' end of mRNA. So bila ribosome nampak, oh adalah tempat untuk saya melekat nanti untuk translate. Dia macam tu lah. Okay, tu dia punya advantage lah untuk kenapa dia modified lah. Okay, faham semua?
Faham, Madam.
Madam.
Okay, good. Bagus kelas ni, semua faham. Okay, nanti saya tak expect you faham 100%. If you can understand about 70-60%, it good. Good enough. So balik nanti you baca lagi. Okay, saya just enzyme saja. Okay, memangkin tindak balas to speed up the rate of reaction without being consumed. Okay, saya speed up. Maksudnya kalau you baca nak faham 1 jam, mungkin you baca 5 minit dah faham lepas ni. Okay, so saya enzyme saja. So you kalau nak berjaya, you lah, bukan saya Kalau you dapat A pun bukan saya, sumbangkan sikit saja. Sebahagian besar bergantung pada diri you sendiri sebenarnya. Okay? Sudah habis dah dekat situ. So kita dah habis transcription process. Kita nak masuk translation process. Okay, saya tak tahu, you perlukan rehat tak 5 minit ke? You perlukan rehat tak sebelum saya masuk translation? Or you want to proceed?
Nak take 5.
Boleh
You
juga
nak take 5 kan? Okey lah, pergilah minum air ke, nak goyang-goyang badan gerak kiri kanan ke. Rehat sekejap, 5 minit saya bagi after 5 minutes kita, sekejap, 1:10. 1 suku kita masuk balik. Boleh
Boleh.
Boleh, Madam.
Pergi minum air dulu. Okay kelas, so boleh proceed ke tak ni? Semua dah masuk ke, dah duduk dah ke di hadapan komputer masing-masing? Depan laptop, depan handphone lah? Dah, betul Tuan-tuan orang ambil bantal dah tidur dah. Selamat dah pun. Okay. Saya sambungkan. Okay, ni kita nak masuk translation sekejap je kelas. Tak lama dan dia tak susah. But you kena fokus lah. Okay, kelas. Ini tRNA structure. The 2D structure. Okay, tRNA. Kalau ingat lagi lukisan saya. Tadi saya ingatkan saya lukis macam mana kan? Okay, saya lukis gambar apa? Penyapu kan? Dah tak ada dah. Sekejap Saya punya pen. Okay. Okay, saya lukis penyapu tadi kan? Bentuk macam ni. Amino acid attachment site. And also this is site untuk anti-codon yang akan complement with the codon. You can see here, this is anti-codon. Okay, anti-codon boleh berubah depends on the codon tu. And then this is amino acid attachment site. Okay, tempat di mana amino acid tu melekat. Okay, saya lukis penyapu je. Kalau dia suruh lukis, saya tak pernah lagi lah dia suruh lukis. Okay, now tRNA tadi, transfer RNA untuk transfer apa tadi? Amino acid, right? Transfer amino acid to codon and ribosomes. Remember? Kalau you ingat lagi kan, dia akan pergi dekat tRNA akan complement with the codon. Codon, ada banyak codon. Okay, and ribosome ni dia akan melekat dekat ribosome sekali. Okay, so actually RNA, okay, are transcribed from DNA template in the nucleus. Okay, for your information class, all RNA, tRNA ke, mRNA ke, rRNA ke, semua okay, they are produced from DNA template juga. So maksudnya ada gene, okay, there is gene that produce tRNA. Okay, so nampak tak? Walaupun kita nampak RNA tu dalam cytoplasm, but actually dia berasal daripada nukleus sebab dia perlukan DNA template. Okay, there is specific gene yang produce tRNA and dia akan travel to cytoplasm because dekat situ berlaku translation process. It is a single strand because RNA kan. Okay, function tadi to transfer amino acids to a ribosomes. Okay. Anticodon akan bind with codon in mRNA and then ribosome akan bind together. Okay, has two sites that I mentioned. There are the amino acid attachment site and also anticodon. Anticodon tu unique for each tRNA. Anticodon ni untuk, so you can imagine we have a lot of tRNA sebab dia unique. Okay, so we have a lot of tRNA specific. Dia akan carry specific amino acid. And each tRNA can be used repeatedly. Okay, this is just introduction untuk bahan-bahan translation. Okay, ingredients to make translation process. Next, you akan belajar, okay, ni dia tunjuk lagi structure tRNA. Okay, macam ni ni anticodon. Okay, kenapa 35? Remember, mRNA 5'3". Okay, so ini yang you akan dapat lah. Okay, so amino acid attachment site allows each tRNA to carry specific amino acid anticodon 3'5". So mRNA 5'3" direction. Class, take note nanti you buat lab dengan Puan Mazhani, dia minta you cari yang table tadi. You see amino acid jenis apa, jangan guna anticodon. Ni silly mistake, student suka buat. Okay, mRNA yang contain information about DNA. So, you bila nak tengok protein apa yang you see for what type of amino acid, you have to look at mRNA codon, bukan anticodon. Okay, faham Tengok dekat mRNA codon. Jangan buat simple mistake. Okay, your first lab, you akan dapat sequence, banyak sequence nanti kan. So you akan cari anticodon dia, cari protein dia. Kelas, jangan buat mistake. You dah boleh dapat full mark untuk your first lab. Sebenarnya you buat complement saja. Ni je yang you akan buat nanti. So don't make simple mistake. Okay? Tengok mRNA. Next, yang ni don't worry, nampak macam complicated, tak payah baca. I just want to tell you that, okay, this is tRNA, remember? And this is enzyme. Okay, nama enzyme ni lah. Enzyme ni dia buat apa kelas? Dia akan make bond between amino acid and this tRNA. Nampak tak? Dia catalyze covalent bonding, which covalent bonding between amino acid here and also tRNA. Tu je sebenarnya dia nak cakap gambar ni. Okay, faham saya just simplifykan je. Okay, enzyme will catalyze the formation of bond between amino acids and tRNA. Okay, tu je, tak ada apa. Okay, now ribosomes. Okay, you dah belajar kan ribosome, protein synthesis protein, whatever kan. You belajar. Okay, it consists of small subunit and also large subunit. As I mentioned before, okay, ribosome is made of proteins and rRNA. Okay, tu komponen utama ribosomes. Okay, now dia pun ada binding site. Okay, tadi saya dah mention mRNA, it is 5'3" kan. Dekat sini untranscribed region ni ada binding site for ribosome. So mRNA pun ada binding site for mRNA. Dia dah sedia dah. So nanti dia akan bind macam ni. So this is mRNA. Okay, dia akan bind dekat here. mRNA binding site. Okay, so dia akan bind dekat sini lah. Okay, sama-sama bind. So dekat sini kelas, in this ribosome ada binding site for mRNA and also there are 3 binding site for tRNA. Nanti later on you akan nampak tRNA akan masuk dekat A site here, carry amino acid. Dia penyapu tadi. Sama juga tRNA carry amino acid here. Okay, sebab ni adalah site untuk tRNA. 3 binding site for tRNA. E site, P site and A site. Okay, A site, okay, A site carry, okay, you jangan tengok lagi kat sini ya. A site dia akan carry, dia akan receive, dia akan ready to receive amino acid, receive tRNA. So kita lagi kat sini, bayangkan, okay, A site is ready to receive tRNA. Okay, that carry specific amino acid. Okay, A ni kosong. Bayangkan dia kosong, dia akan ready to receive tRNA with amino acid. P site, okay, P site, dia akan hold the growing polypeptide chain. So this is tRNA, this is the growing polypeptide chain. The amino acid. So you have polypeptide here. So dia akan hold, dia akan pegang, hold the growing polypeptide chain. Sekarang you tak nampak lagi kelas. After this you akan faham. Student takkan faham di sini. Okay, I'm sure. Semua you kena tahu function dia dulu. Now the E site is where tRNA leave the site. So dia akan, tRNA tu gedek-gedek keluar. Dia keluar daripada sini. Keluar. Okay. So yang kat sini just penerangan. You tak akan faham the process. Now kalau you tengok proses, you akan faham. Okay. Now saya nak mulakan proses translation. Stay focused. Translation, you want to make polypeptide chain. There are 3 stages here, same as transcription, which is initiation here, first stage. Next we have elongation and termination. This is your mRNA that runs 5'3". Okay, tengok tRNA ni. Nampak tak? Okay, this is A site. Okay, ni dekat A site here. This is A site. This is P site. This is E site. Okay, nampak tak? You imagine tadi A site kosong, next amino acid to be added. Dia kosong, dia akan ready to receive the next amino acid. The next tRNA with amino acid. Okay? And dekat sini kalau nampak growing polypeptide dekat sini. Now, saya nak you tengok proses dia. Now, tengok fokus betul-betul. Okay, we have mRNA here. Okay. Initiation, permulaan, Okay, the first tRNA kita panggil initiator tRNA. It has anticodon here and it has codon in here. Start codon. Okay, start codon tadi saya dah ajar. Start codon is AUG. Maksudnya if you have mRNA 53, you have here AUG. Bila dia nampak base ni je, okay, CUG. Okay, bila dia nampak AUG ni, this is the start codon. Maksudnya translation process will begin here. Okay, so apa yang berlaku? tRNA. Kalau A, anticodon dia apa? UAC. Right? This is the anticodon. Tapi kalau you nak tengok amino acid apa, you tengok dekat AUG. AUG carry amino acid apa? Methionine. Okay, so nampak ni MET. So first tRNA will enter the P site. Dia akan masuk dekat P site. Boleh imagine tadi? APE. First initiator tRNA will enter the P site. Carry amino acid MET. Okay? Nampak ni. Initiator tRNA carry MET attached to codon. AUG. Dia find dekat sini. So you will imagine here. Okay? And you have small subunit before that. So after process ni apa jadi? The last subunit akan come. Debak datang kat situ. Okay, selepas dia bind dekat sini, so dia akan form translation initiation complex. Okay, nampak tak kompleks? Sebab apa? Sebab dia ada small LR subunit, dia ada mRNA and dia ada initiator tRNA and dia enter dekat P site class. Okay, what happened to the vacant A site? Vacant A site is ready to receive next tRNA with amino acid. Okay, so dia panggil amino acyl tRNA. Dia nak cakap yang tRNA tu ada amino acid. So dia panggil amino acyl tRNA. So vacant A site is ready to receive next amino acid, next tRNA with amino acid. So dekat sini dah ready dah. Dengan next amino acid, dia akan masuk dekat A site. Ready. Dekat kosong dekat situ. Now, kita tengok next. Okay, class, saya nak you, okay, under elongation process, tadi initiation under elongation process, we have 3 stages actually. Codon recognition, peptide bond formation, and also translocation. Saya akan terangkan, dia tak susah. Ni je, lepas tu habis dah. Okay, class, tengok betul-betul kat sini. Can you imagine, yang ni saya nak padamkan. Okay, this is MET. Sebab kita belajar MET tadi kan. MET will enter at the P site. A, ready to receive next tRNA with amino acid. Okay. So apa jadi? mRNA codon in the A site will form hydrogen bond with anticodon. Betul tak? mRNA, contoh tadi. So CGU. Okay. So the next tRNA, anticodon dia apa? GCA. Dia akan form hydrogen bond here with codon. Anticodon with codon will form hydrogen bond. Okay. Of incoming amino acid tRNA. So it will enter the A site. Itu adalah codon recognition. Maksudnya tRNA with amino acid will enter at the A site. Tu je. Okay. Next. Apa jadi kelas? Next stage. So tadi saya nak buang yang ni kelas. Okay, saya tak nak you confused. You imagine this is MET. This is MET dekat P site. Now dia dah masuk kelas. Dia dah masuk here. You nampak ni with the next amino acid. Next step, what happened? Peptide bond formation. Enzyme, okay, this enzyme will catalyze the formation of peptide bond, okay, between amino acid at the P site to the new amino acid in the A site. So you nampak tak ni? What happened to this one? Yang ni dia akan pindah pergi sini. Tak. Dia akan make peptide bond dekat sini between amino acid at the P site and amino acid at the A site. So you akan dapat saya potong yang ni. Confused? Kalau you nampak tak? Peptide bond formation here. This one. Dia berlaku dekat A site. What happened to the P site? Dah putus dah dekat sini. Break. Okay, so the polypeptide separate from the tRNA. Nampak tak? Dia separate tadi, MAT lah. MAT dalam contoh saya bagi MAT lah sebab saya dah padam. MAT separate from the tRNA in the P site and bind to the amino acid of the tRNA in the A site. Oh, dia dah bind. Okay, next. Apa pula seterusnya berlaku? Okay. Now, you bayangkan, okay, ni saya tak ada tadi. Okay. So, apa yang jadi? Can you imagine yang A site ni dia akan move to the P site. Yang P site akan move to the E site. Di sini nampak tak ni? A masuk dekat P. P masuk dekat E. Nampak tak? Memang nampak pun slide kan? So, apa jadi dekat sini? Okay, what happened? So, dekat sini, you nampak tak function P site? P site holds the growing polypeptide. That's why dia kata P site function dia holds, pegang, holds the growing polypeptide. Yang tadi yang ni next tRNA tadi dia akan masuk dekat E site. So bila masuk dekat E site exit. So dia akan leave, keluar. Okay, yang A site ni sentiasa akan jadi kosong to receive next amino acid, to receive next tRNA with amino acid. Gerek-gerek datang. Maksudnya dia akan sentiasa bergerak ke depan sini sebab dia nak translate. Okay, ribosom akan move forward from 5 to 3N. Okay, sebab dia nak translate. So, translocation ni maksud dia, tRNA in the A site translocate to the P site. Okay, dan dia akan bawa sekali dan dia akan tarik sekali mRNA. So, you bayangkan mRNA ini akan bergerak ke depan. Bergerak. So, yang di hadapan ni kelas, kodon yang di hadapan ni, kodon, apa jadi kat kodon di hadapan? Dia akan masuk dekat A site here. Bila dah masuk dekat A site, it will be translated. Okay, so this bring the next codon to be translated into the A site. So the previous tRNA in the P site will enter the E site and release from the ribosome. So it proceed in a 5-3 direction. Faham ke tak? Can you imagine? Cepat gerak. Ya.
Macam tu juga lah.
mRNA along with it tu macam mana?
Yang ni kan dia memerlukan you imagine sebenarnya. Kalau you boleh imagine kan, dia macam melekat tau ni, dia melekat. Dia akan, bila dia bergerak, mRNA yang kat sini pun akan bergerak ke belakang. Boleh bayangkan tak?
Dia ke kiri lah.
Dia, yes, ke kiri. Dia akan masuk sekali dengan kodon. Yang daripada A site tadi dia akan masuk ke P site. Dia akan bawa sekali mRNA ke belakang. So dia akan tarik sekali. So yang kodon yang dekat sini ni, dia depan A site tadi akan masuk pula dekat A site. Kodon yang baru akan masuk A site. That's why dia akan translate kat sini pula. So sebab tu A site, tadi saya mention function depan-depan, always ready to receive next tRNA. P site hold the growing polypeptide. E exit ataupun leave. Certain textbook kalau dia buat dua je site, dia bagi A site dengan P site sahaja kelas. E tu is macam imaginary site. Bila dia masuk je ke sini dia dah keluar. Boleh faham ke tak yang kat situ? Dah habis dah sebenarnya translation ni. Yeah, is it? Yes. Sebenarnya okay, last step termination. Saya nak habiskan sebab penat nanti. Okay, termination. Class remember, AUG start codon. Okay, bila dia RNA polymerase tadi dia transcribe, okay, bila dia jumpa codon dekat mRNA, dia jumpa UAG ke UAA ke UGA ke, dia akan stop the process. Stop codon, stop. So tak ada tRNA yang akan bind dekat sini, dekat A site here. So dia akan gantikan dengan release factor, okay, protein. Okay, so release factor will bind at the A site here. So apa jadi? Okay, release factor protein bind to the A site. Dia akan hydrolyze the bond between tRNA in the P site and the last amino acid. And dia akan release polypeptide. So you nampak tak apa dia buat? Dia akan menyebabkan ni. Nampak tak? Polypeptide ni dia akan separate from this tRNA. Dia akan hydrolyze the bond here. Okay, between tRNA in the P site and the last amino acid here. And then apa jadi? Dia akan release the free polypeptide. And sama juga the two ribosomal subunit, the large and the small and other components here will dissociate. So, translation process pun habis, finish, the end of the story. Tu je sebenarnya. Okay, so yang ni kena ingatlah UAG, UAA, UGA is a stop codon. Okay, so dia tak code for amino acid, but it acts as a signal to stop translation process. Okay, saya ada okay, class for your information, lagi saya tunjuk video, hopefully dia boleh summary, dia boleh summarizekan kita punya apa ni, transcription and translation. But for your information, tadi saya cerita hanya one story, but actually mRNA ni sebenarnya ada banyak ribosome actually, so dia akan speed up the process actually. Okay, dia akan speed up the translation process. Tak ada apa pun. Okay, yang ni make sure you differentiate between prokaryote and eukaryote. Transcription macam this is eukaryote because transcription occur in the nucleus, and dia akan produce pre-mRNA, and dia akan process by adding cap and also poly-A tail, and dia akan export out, and this is good sebab dia nak tadi ada 3 advantages tadi kan, yang dia nak facilitate the movement from nucleus to cytoplasm, dia nak protect from hydrolytic enzyme, and dia boleh menyebabkan ribosome kenal tahu kat mana nak attach. Okay, so now bila dia dah translate at the ribosomes, you nampak tak A site, P site and E site. A site always ready to receive next tRNA with amino acid. This site holds the growing polypeptide, and at the end of the reaction, ni akan break. The bond will break because dekat sini tadi release factor akan bind dekat A site. Bila dia jumpa stop codon. Okay, so that is translation lah, and dia berlaku dalam cytoplasm. Okay, I will show you a video. Yang ni saya takkan masuk. I will show you video. Okay, dia akan summary kan transcription and translation. At least you dapat tak general idea what is transcription and what is translation. I'm sorry kena ajar laju sebab yelah, DNA ni memang kena ajar laju supaya kita sempat nak kejar. Ada banyak lagi kan, saya tak sempat masuk gene regulation. Okay ke semua? Dapat? Okay kan? Okay, tengah hari. Saya nak tunjuk video. Okay, kejap ya. Saya ada dia. Okay, semua fokus. 6 minit je video ni. After that, we end our class. Okay? Saya stop presenting sekejap. Okay, ready semua? Nampak tak?
Tak nampak tu.
Nampak. Tak nampak.
Nampak? Dengar kan? Kejap ya. Oh, error. Okay, dismiss kejap ya. Can't share. Nampak tak?
Transcription and translation.
Nampak ke tak?
Nampak Adam.
Nampak ni dah. Nampak, okay.
Nampak dia.
Thank you. Sekejap. Okay, fokus 6 minit. Lepas tu kita
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish or a cat or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene. And after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this 2-step process, DNA is transcribed into an mRNA, and then this mRNA is translated into a protein, all simply by obeying the base pairing that occurs in nucleic acids. And since every gene codes for a specific protein, and proteins make up most of what you are, from your muscle tissue and organ tissue to all of your receptors and enzymes, this is how DNA carries the code for a living organism. Thanks for watching, guys. Subscribe to my channel for more tutorials, and as always, feel free to email me, ProfessorDaveExplains@gmail.com.
Okay, faham semua? Okay ke semua?
Faham, madam. Okay.
Kalau you faham video tu maksudnya faham lah penerangan saya yang sangat laju tadi. Saya terpaksa laju sebab nampak 1:45 dah pun. Okay, kalau apa-apa tak faham bolehlah email Profesor tadi Okay, so tak ada soalan I assume tak ada soalan. So go back, nanti balik semula, tengok balik Canva, whatever Solomon ke ataupun internet ke, you baca, saya rasa lebih mudah lah you faham lepas you lepas attend kelas ni. Okay kelas, semua okay ya? Terima kasih semua, thank you. Okay, jumpa lagi nanti. Ya,
Thank you, madam. Thank you, madam.
ada. Okay. Okay.
Thank you, madam.
Thank you, madam.
Thank you, madam.
Okay.
Thank you, madam.
Madam.
Welcome.
Thank you, madam.
Welcome. Welcome. Okay.
Madam,